How can cells with the exact same DNA in their nuclei perform different functions? For example, what makes a muscle cell different from a skin or epithelial cell if they all have the same DNA? Well, this is because cells express different genes and therefore different proteins. Not every gene on the chromosomal DNA is turned on or expressed. The cell expresses genes based on a number of different cellular requirements. This is called control of gene expression. When a gene is turned on, protein synthesis begins. A gene expresses its intended protein through transcription and translation. When a gene is turned off, protein is not made. Therefore, two cells that have the same set of genes can express very different proteins based on the function of that cell and which genes need to be turned on. For example, muscle cells turn on genes for actin and myosin production, whereas skin cells turn on genes for melanin production. Because control of gene expression in eukaryotic cells is so complex, we're going to focus our discussion on control of gene expression in prokaryotes. Prokaryotes cluster related genes together and put them under the control of a single promoter so that they are transcribed into a single mRNA molecule by RNA polymerase. Operon is the term given to this cluster of genes. The promoter region is where RNA polymerase binds to begin transcription of the DNA template strand into messenger RNA. Also notice that right in the middle of the promoter region is an operator region. The operator region can bind a repressor protein that will block RNA polymerase from passing and transcribing the genes downstream in the operon. The most common example of this operon model is the lac operon found in bacteria. Lactose is a nutrient that is metabolized by bacterial cells as a source of glucose. There are 3 genes in the operon, and they are called LAC-Z, LAC-Y, and LAC-A. The proteins that are encoded by these genes transport and break down lactose. If lactose is not present, the repressor protein binds to the operator region, and RNA polymerase cannot pass. Therefore, the mRNA for LAC-Z, LAC-Y, and LAC-A are not transcribed. However, if lactose is present, it will bind to the repressor protein, inducing it to change its shape and fall off of the operator region. RNA polymerase is now free to pass and will transcribe these 3 genes into mRNA so that the ribosome can make the proteins for transporting and breaking down lactose. Bacterial cells are very efficient and use operon models to tightly regulate gene expression in response to their environment and nutrient requirements. By switching genes off when they are not needed, cells can prevent resources from being wasted.